our second to last video for Botany 105. These last two videos will be focusing on organisms that we refer to as fungi. Sometimes you hear that word pronounced fungi. They're both correct. You'll hear even experts pronounce them both ways. So this is going to be part one, where we're going to really kind of talk about the introduction to these organisms. And we're going to talk about the kind of most well-known group of organisms that we generally refer to as mushrooms. And those are the mushrooms found in Basidiomycota. So the learning goals of this video: to learn the phylogenetic relationships between the true fungi, plants, and animals, including yourself; to learn the generalized morphology of a typical mushroom; to understand how fungi in the phylum Basidiomycota reproduce; and to know the similarities and differences between the life cycles of fungi and plants. And then, just so you know, in the next video, we'll be learning about some other types of true fungi, as well as some fungus-like organisms, so things that we kind of refer to as fungi, but they're not true fungi. So, talk about what that means here. Here's a list of your vocabulary. Feel free to pause it and copy these down. It's just nice to have. They're not necessarily in order. Moving on. So this is a sort of pixelated giant phylogeny that's sort of in a circle here, showing the relationships between a whole bunch of different organisms that we've talked about this semester, and many that we haven't, many that we won't. So up here are all of our sort of our true plants. We've got the algae, the red algae, the land plants, right, the streptophytes. But then it's way down here that we've got the true fungi. Way down sort of from this clade right here. And notice that you are nestled right next to them relative to these other groups of eukaryotes. We are actually very closely related to fungi. So we're more closely related to fungi than fungi are to plants. So in other words, if we were to look at a cladogram of the relationship between plants, P for plants, and then I'm going to write you right here, humans, animals, in other words, and then fungi, F for fungi right here. Notice that, fungi are more closely related to you than they are to plants. And that might come as a surprise. You think of fungi as sort of these stationary organisms, and they're very similar to plants in that way. Anyway, some fungal origins, they really are not completely sure how long ago fungi evolved, maybe 1 or 2 billion years ago. The Paleozoic era, about 540 to 250 million years ago, fungi were mostly aquatic. But they first showed up on land around 460 million years ago. And there's lots of fungi fossils from 410 to 360 million years ago. And these fossils are actually, they come in a few forms. Some stems, so stems of plants with fungal hyphae inside of them. Also, spores are really good at fossilization. Okay, the importance of fungus. Well, there's tons of it. First of all, forage. And the first thing I'm going to say about that is, whenever you do forage for mushrooms, it is so important that you are confident with your identification of these things, because there are mushrooms that are deadly as well, and they look almost exactly like some delicious edible mushrooms. And so it's good to be either with an expert, or to really have taken sort of an identification class or something like that before you just go eating mushrooms from the wild. But morels are the ones that you see up here in the top left. Those are delicious local edibles. We have many of those, especially this time of year. Well, not morels, but other types of delicious mushrooms this time of year. Well, some other kind of less well-known relationships between humans and fungus comes from things like a member of a fungus called Penicillium. And this, of course, is responsible for the antibiotic penicillin, which has saved countless lives around the world. Well, other important things, lichenized fungi, we're going to talk about those in the next video. Mycorrhizae, these are the relationships between fungi and plants. Yeast, fermentation, again, food for animals and humans. Also, lots and lots of medical conditions come from fungi. In fact, there are about 300 species of fungi that are pathogenic to humans, and about 8,000 that are detrimental to plants, and therefore indirectly to humans. Okay. So, kingdom fungi is sort of a term that's a little outdated, that term kingdom. Mycology is the study of the true fungi. Okay, and here is a picture of turkey tails on the left. This is a local mushroom that you can find if you probably go walk around Humboldt a little bit maybe in the woods. These tend to grow on hardwoods, and though not edible, these have medicinal properties to them. And again, before you go using any mushrooms in the wild for medicinal purposes, even you've got to really kind of do a lot of research and see exactly kind of how to extract the medicinal properties, and make sure that you've got the right thing. That's the most important. Here's a picture of Holmes helping me sniff out some mushrooms. He knows to not eat any, for sure. And then even down here, these are some really interesting looking, really young mushrooms. Well, there are about 100,000 described species of mushrooms, and new species of mushrooms are described, I mean, literally all the time. You find new publications maybe every week or month of many new species of mushrooms being found. And the truth is that scientists believe that there's about 5 to 10 million species. So many of the species are just unknown. Maybe it's because they're so small, they live in such specific habitats. Who knows? Okay, so let's talk about kingdom fungi. What do they all have in common? Well, they all have mycelium made of hyphae. So what is that? Well, a mycelium is sort of a mass of hyphae. And a hypha is sort of one of these filaments. Okay? So like we've got a hypha over here, we've got a hypha over here. Together, all of these kind of branching filamentous structures, the hyphae, are called mycelium. So here's a actual picture of a scanning electron microscope image of a hypha, or some hyphae rather. Sort of notice how they're all kind of branching off of one another. Okay, that's what hyphae do. And you probably have actually seen these if you've ever accidentally let your fruit go moldy. You get this sort of stringy stuff that grows all over it. Or maybe you've noticed it on wood or in soil. Yeah, it's kind of the stringy filamentous stuff. And hyphae, remember a hypha is a singular kind of filament. It emerges from a spore. So they do use spores kind of similar to other organisms we've talked about to reproduce. And we'll talk about what those where those spores come from here in a little bit. So kingdom fungi. These are multicellular organisms. We don't have any unicellular organisms in this kingdom. And they're always going to have that filamentous body plan, at least to some degree. Now, notice this isn't exactly filamentous, but it has a filamentous part of its life cycle. It's namely, it's underground. Okay. And these are also heterotrophs. These are not photoautotrophs like all of the other organisms we've talked about this semester. These things actually do not produce their own food. Instead, they're more like me and you. They have to sort of eat their own food in a way. Now, they don't eat it the same way we do, but they are heterotrophs just like us, meaning they have to get their food from something else, as opposed to making it themselves. The autotrophs, they make it themselves by using sunlight, not fungi. So what do we mean by this filamentous body plan and being heterotrophic? Well, what they do is they feed by surface absorption. With all of these filaments in this massive mass, since instead of them all being clumped together like you and I, all of our cells are just kind of in this one giant clump, all of their cells are spread out in this mass of filaments. And so they have a massive surface area. And they're able to produce these enzymes that they sort of are able to release from all of their hyphae. And they can break down the kind of the material that's in their environment and feed on that by absorbing it through their massive surface area. So these are decomposers. What they're not feeding on the same type of stuff that you and I eat. Instead, they are feeding on usually dead stuff. They're saprotrophs. And other times they're parasites or pathogens. So sometimes they do feed on sort of live things and they'll maybe slowly kill them from the inside. That happens sometimes. But a lot of times fungi are saprotrophs, meaning they are kind of eating, dead stuff, decomposing material. Or decaying material, rather. And that's what we mean by they are decomposers. They're breaking down those They're breaking down those dead kind of the dead matter that's in the environment, animals, plants, and they're breaking them down from those complex molecules into simpler constituents, ones that can be uptaken by plants and animals. And then they can kind of, those smaller constituents can then be rebuilt. And this, of course, is extremely ecologically important. It prevents massive buildup of, dead stuff. So they are extremely, extremely important ecologically. We could not live without them. Nothing could. Now, they're either decomposers or they're mutualists. They can come in the form of lichens or mycorrhizal associations, and we're going to talk about both of those. Okay, now they do have a haplontic life cycle, some with a dikaryotic phase. So we're going to define this term dikaryotic in a sec, something that's really different from any of the plants we have talked about so far. But remember, haplontic means they have a multicellular haploid stage. In fact, they do not have a multicellular diploid stage. So that diploid structure, the zygote that is formed, it will immediately undergo meiosis. So to be clear, they do have a life cycle, just like plants, in the sense that they will undergo fertilization, form a zygote. That zygote will then form some haploid structures, and those will undergo mitosis. Yeah, it's kind of amazing how similar they are in their life cycle. They have chitin in their cell wall, and this is really important because chitin is a substance that's found in animals, not all animals, but a lot of animals, especially some sort of evolutionary ancestral ones. And so this is one of those features that fungi have in common with animals. This is one of those signs of evidence that points to the closer relationship between animals, closer than plants, that is. Okay, so in terms of the diversity of fungi, there are kind of at least five well-known phyla, but the phylogenetics are being clarified by genetics really frequently. And so this may or may not change. But anyway, we're only going to be talking about two different phyla. We're going to talk about Ascomycota and Basidiomycota. And just so you know, there's actually a class at HSU called Ascomycota and Basidiomycota, and it's taught as a 300-level botany class. And it's a really cool course that's offered if you do want to expand on this topic. We are just going to brush the surface of these two groups. And yeah, the Basidiomycetes are kind of your typical mushroom with a cap shape. And then the Ascomycetes are sort of the cup fungi. We're only going to be talking about these two. And actually in this video, I'm only going to be talking about this one. Ascomycetes will be in the next video. So there are also two sort of informal groups of fungi that these are both going to be talked about in the next video. Deuteromycetes and yeasts. And the two types of mutualistic symbiosis that I sort of mentioned earlier is lichen symbiosis, which we'll talk about in the next video, and a mycorrhizal symbiosis, which we'll talk about in this video. Okay. So actually we'll talk about that right now. The mycorrhizal associations between fungi and plants are found in the roots. Okay, so a mycorrhizae is a hypha that's associated with a plant, or a mycelium that's associated with the plant. And this intimate association really helps out both parties, both the plant and the fungus. And so what we see here, this is sort of like a pie cutting of a root. Imagine this whole circular kind of root underground, and in between some of these cells, right, we've got these black, and now I'm coloring them over in red, filaments. Well, that's the fungus, and it's really embedded itself within the plant root, in between the cells in this case, and in fact even inside of the cells in some of these. Okay. So mycorrhizal associations are, my goodness, they are extremely ecologically important. Without these associations, plants would not grow to the degree that they grow. Virtually every single plant on Earth has an underground association with at least one fungus, usually many. And if you remove those mutualistic fungi, then the plant doesn't grow as much. So notice these two images. On the top is a set of plants. This looks like some sort of cedar that was grown in a sort of a sterile inoculated, or in a sterile setting. And down here, the soil was inoculated with their mycorrhizal associates. And notice that the plant just grows way faster, way more in the same amount of time when it does have these mycorrhizal associations. And, oops, here we go. When we look at a plant underground that's been growing for a while, really the plant's roots are just sort of these darker brown things. They don't extend that far. All of this other extremely intricately netted massive filaments, this is all the fungus, but it's all connected to the plant, and it kind of extends the ability of the plant to absorb water, right? So underground, this is what they're doing. They're absorbing water, just like we know that's what plants do, and really the fungus allows it to absorb water more efficiently. Okay. And so that's what's happening. For the plant, they're getting more water. They're also having better access to essential elements, namely things like phosphorus, nitrogen. The benefit to the fungus, so what the fungus is getting in exchange is, well, sugars, right? That's what the plant is so good at doing. It makes sugars on its own. It's autotrophic, right? And it's doing photosynthesis all the time. It's producing those sugars. And, well, the fungus is sort of stealing some of those sugars, but it's not a one-sided steal, as we're noticing here. That is what makes this a mutualistic relationship. They are both getting a benefit. So sometimes those hyphae are outside of the cells, as in ectomycorrhizal fungi. Sort of notice that here the hypha is in between the cells. In an endomycorrhizal relationship, those hyphae literally go inside of the cell. So the cell membrane of the plant cell will literally invaginate, and the hyphae will branch, and it will end up inside of the cell. So much more intimate. So ectomycorrhizae might sort of look like this under the microscope. These sort of kind of thicker lines here, these are those fungal relationships. They notice they don't go sort of all the way in, deep inside of the plant root all the time, but they're in between the cells, and a lot of times basidiomycetes will do that. We'll talk about what those are. So this is a cross-section of a root plant, and on the outside we have a whole lot of layers of this mycelium. It's really wrapped itself around the root, and that's why sometimes these roots will look like they've got this kind of chunky stuff on the outside. Well, this is the relationship that the fungus has with plants. It will really become embedded around, it'll kind of embed itself around the root, and sort of increase really the surface area of the root. Okay, so endomycorrhizae, on the other hand, again, it's when a hypha will embed itself within a cell. So that's, if you follow this red here, that's what's going on here. Now the fungus has made itself inside of the cell, and it's branching all inside. And now actually the surface area is even greater between the connection between the plant and the fungus. Cool. Down here, we have an actual picture of what it would look like in a microscope after a cell has been nicely cross-sectioned or longitudinally sectioned with a fine razor and then dyed. So these are not natural colors. And so these lines here, these sort of brick-like cells, these are the plant cells. And then the darker lines here, these are the fungal cells. And if we zoom in on say one of these right there, that looks like it's got that darker structure, that's what this is here. They've zoomed in on that, and wow, look at all of those branching, networked hyphae that are within the cell of the plant. That's amazing. So that's endomycorrhizae. That prefix endo literally means inside, and ecto means outside. Okay, so now let's talk about the organisms themselves, Basidiomycetes, informally, but they are in the phylum Basidiomycota. So that suffix mycota always means a mushroom. I mean, it always means a member of, well, it's actually sort of an informal thing. It used to really sort of imply that you were looking at a member of the true fungi, but as we're going to see in the next video, there are some organisms that have that same ending, but it's because they're fungus-like. So again, mycology, it's the study of fungi. That myc, it really just implies sort of fungus. Okay, anyway. What we're seeing here are the cells of their hyphae, sort of underground here. Like this is one hypha. And they have this really interesting thing. All the members of Basidiomycota, they have this thing here called a clamp connection. I'll come back to this in a sec. And it's this sort of lump in between hyphal cells. And it sort of might look like this too. This is one that's forming here. Well, they have to do this because their cells are dikaryotic. This is wild. Dikaryotic cell, it's not technically haploid nor diploid. Remember, a haploid cell is a cell that has just one entire set of genes. And a diploid cell is a cell that has two entire sets of genes. So all of our human cells, they're all diploid. They all have two sets of genes, except for our sperm and egg cells. Those are haploid. They have the one set. And here, what they have is a single cell that has two separate haploid nuclei. That's wild. So like this is one cell right here, and it's got two nuclei in this one cell. What? They're each haploid, and in theory, they're different from one another. That's why they have them in a different color here. They're not necessarily the same. Now, it's the same set of genes, but they're not identical alleles within those sets of genes. This is wild. Crazy. So in order to, we really aren't going to talk about the details here. I'm not going to force you to know why a clamp connection is formed, but it is quite interesting. Since they want to, when they're trying to grow, each of these haploid nuclei need to undergo mitosis, and in order to sort of have the space for it, and in order to sort of separate the two, one of them has to go into this other side so that a break can form here, and now we can end up with two nuclei or kind of one type of each nuclei in the new cell. It's wild. You don't need to know those details. But you do need to know what a dikaryotic cell is, because you end up with these two dikaryotic cells, and then all of their cells end up being dikaryotic in at least some of their hyphae. Now, remember earlier I said they have multicellular haploid hyphae. Yes, they do. They're either going to have haploid hyphae or dikaryotic hyphae. And here's the difference. A dikaryotic hyphae is one that's going to become, reproductive. Now, there's a lot of fine details here. Let's just get at the really important stuff of what you need to know. Okay, already talked about what dikaryotic is. And this represents a hypha with 3 dikaryotic cells. You see that? We've got 1, 2, 3 dikaryotic cells. Each of those cells has 2 nuclei inside of it. Each of those nuclei are haploid. So we have like this one black haploid nuclei, one black haploid nucleus and a greenish, bluish haploid nucleus. Again, right here in each of those 3 cells. Okay, so karyogamy and plasmogamy are 2 different sort of phases of fertilization. So remember, in all the life cycles we've talked about previously, we have just meiosis and then fertilization. Those are the 2 big life events. Meiosis makes a cell go from diploid to haploid. Fertilization makes a cell go from haploid to diploid. Well, they still do have meiosis, and meiosis is exactly the same. It's when you've got a diploid cell that goes to haploid. But in order to get that diploid cell, the nuclei must fuse, right? So that these 2 nuclei right here will fuse, and that's called karyogamy. Karyogamy is when nuclei fuse, and they result in a diploid cell, okay? But if you go back a step now, like how did that dikaryotic cell even form? Because most of the cells, let me go back here, in a hypha are actually haploid. So like here we have a haploid hypha. Each of the cells only have this one nucleus. Okay, same with over here. Each cell only has 1 nucleus. So in order to get a cell with, 2 separate nuclei, plasmogamy must occur. Oops, sorry, that. Let's see if I can fix that. Nope, I can't. Plasmogamy is the protoplasm of the cell fuses, but not the nucleus. So the protoplasm, that's just essentially everything inside of the cell wall inside of the cell membrane that is not the nucleus. Okay, so everything, the cell fuses, but the nuclei do not fuse. That's plasmogamy. And you end up with a dikaryotic cell that is essentially twice the size. And it's got twice the nuclei, since they did not fuse. Okay, so Basidiomycota. When we look at a sort of a typical mushroom, when you think of a mushroom, it's got the cap, right? That sort of top part. It's got sort of the stalk. But when we look underneath, you might notice there are these things like maybe gills on the underside. And what they've done here is they've done a cross section or a little section there, and then we've zoomed in. And this is one of those gills right there. And now they've done the same thing. They've taken this little tiny section right there, and they've zoomed in. Okay, so what happens is lining all of these gills, all of the underside of these gills, we've got these structures that are producing spores, okay? This is the typical reproductive structure of a member of Basidiomycota. They produce spores. Those spores are called basidiospores, conveniently. And they're produced on the structure that's called a basidium. So that's where those words sort of come from. The basidium is the structure that produces basidiospores. So let's look at that again. So this is another sort of recap, cap, and stalk. Now on the underside here, we've got all these gills. That's what we're looking at under here. And under the microscope, you can actually kind of see here, there are a bunch of those little basidia. So those little sort of structures. A basidium is the structure like this. And it's got these 4 basidiospores. Okay, so this whole thing is a basidium. And one of those basidia, it's very small in this picture, it's like right there. And then there's another one like right there. All the little black structures here, those are the basidiospores. And there's 4 of them per basidium. And look at that. Great. There's another look. So here we've got all those gills. And when we look really closely, lining those gills, we've got the basidia producing basidiospores. Now that layer of basidia and its associated basidiospores is called a hymenium. Okay, so that hymenium is the layer of basidia that is lining the gills. Okay, it's this fine layer right there. Okay, some more terms. Basidiocarp or a basidioma. These are 2 kind of botanical terms for a mushroom. Okay? And a lot of times mushrooms have this kind of typical development. When they're really small, they might be called a little button, and they don't really resemble that typical mushroom look yet. But if we were to slice this open like that, this is what we would see on the inside. Okay, we would see actually a tiny baby mushroom inside of that outer shell. That outer shell is called a universal veil, as shown in red here. And then there would also be this smaller connecting structure called a partial veil, and that would sort of connect the gills to the stalk of this tiny, tiny baby mushroom. Now, this will grow into your typical mushroom with a cap and a stalk, but we have better terms for that, more botanical terms. A pileus is the other kind of botanical term for the cap of a mushroom, and then the stipe. That's the better term, if you will, for a stalk. Okay, so the universal veil and the partial veil end up on that adult, matured mushroom. The universal veil gets kind of pushed up and out, and it ends up being like these little kind of chunks on the outside of the cap. And then the stuff from the bottom, the universal veil that was kind of along the bottom, will remain on the bottom, and it'll become this thing called a volva. Now, the partial veil, which was connecting sort of the end of the gills, the end of the cap to the stalk, the stipe, those are those end up being called the partial veil remnants or the annulus. Now, this picture down here does not have an annulus, but if it did, it would have it right there. And that would be the partial veil remnants. And it looks like we just can't quite see if this picture down here has partial veil remnants attached kind of on the inside underneath there. Okay. Now the hymenophore. Now this is remember how we talked about the hymenium? Same sort of root there. The hymenophore is all of the hymenium. Okay, the hymenium was that tiny microscopic layer of spore-producing structures, the basidia. And the hymenophore is sort of how those layers of hymenium are associated with one another. And it's just sort of usually it's the underside of the cap. And sometimes it's, a lot of times actually, it's gills. It's like this. So you flip over a mushroom like they've done in this picture, right? And then you see all those lines. And under the microscope, if you were to take a really fine section of those, like right through them, well, maybe not that way. I can't really do this two-dimensionally, but maybe you can kind of see the relationship here. This is one cap, right? And so these are all the gills. And again, the hymenium is lining all of these gills. This is just like a little two-dimensional slice of it. And it's that hymenium layer that's shown right here that we've zoomed in on, and that's where you can see the basidia and all of those little basidiospores. Okay, so there's a few different major types of hymenophore. And again, it's sort of the underside of that mushroom. You have to flip it over, and sometimes they can be really surprisingly beautiful underneath, and it's very exciting. Now, when you pick mushrooms, you don't really have to feel bad because it's kind of like picking an apple on a tree. It's sort of the fruiting body of the mushroom. The mycelium will remain underneath, and you'll be spreading the spores, really, by picking these mushrooms. Now, at the same time, it's always nice to leave a mushroom in its natural condition, but if you ever are just, if there's a lot of them nearby, don't hesitate picking one or two and kind of looking at how cool the mushrooms are underneath, or the gills, or whatever the other hymenophore type is underneath. So what are the other types of hymenophore you might see? Well, if you do see gills, the mycological term for that is called lamellate. Okay, and that's shown here in this diagram. It's when you've got what we've been talking about, the typical sort of mushroom gills, kind of lines radiating underneath. Another type you might see is poroid or porate hymenophore. And this is when the hymenium, it's lining the insides of these tiny pores. So in this photo, imagine those tiny basidiospores, they're being produced inside of these tubes. We can't see how deep these tubes go, but imagine they go probably very deep, as you can sort of see here. And there's just, as you can see here, just bajillions of these tiny pores, and just imagine just trillions of spores being produced throughout the lifetime of these mushrooms. Lots and lots of spores. Okay, the third major type of hymenophore you'll see is the adentate hymenophore. This is when they're more or less toothed, sort of pointy underneath, and the hymenium, again, it's just lining all of those teeth. The last type is smooth, and this is where you just kind of have an entirely smooth, no other texture of the hymenophore here. Now, here's a little summary slide showing a few more examples of the gilled, the poroid, the dentate, and the smooth hymenophore types. So how the hymenium layer is sort of arranged. Whoa. Sometimes mushrooms of this family come in really shockingly different forms. They are sometimes known as puffballs, or like down here, there's this really cool one called a bird's nest fungus. Wild things can happen, and we're not going to talk about all of those wild things, but at least need to show you this one thing. Basidiocarps are sometimes not in that typical form of a cap and a stalk. Sometimes they're in this essentially sphere. And what happens is one of those spheres on the inside has this structure. It has this kind of stuff called gleba. And actually sometimes these puffballs are edible. I've eaten many edible puffballs. And they're edible at this stage. Inside it's just sort of one solid mass. And now, just to remind you, not all of them are edible, so do not eat them all. You have to look this up. But it's just like this solid mass, and it makes sense that it's not like gross to eat or anything. And I'm just saying that in light of what happens later. As they mature, that mass that was called a gleba, it becomes this stuff called capitulum and a mass of spores being produced from their basidia. And it just becomes this mass of powder, essentially. And they'll poof out of a little hole in the top. And let's see if this works. Kind of like this. Cool. Nice. So if you ever see one of those, feel free to poof it. And what happens if you don't puff it is little raindrops land on it, and they end up making the spores kind of poof out, or maybe some animal will step on it. Very cool. Here's just a study tool. Really, this has a lot more terms than what you need to know. Just focus on the list of terms from the beginning of this video. This is for your reading pleasure. If you are very interested in this stuff, I highly recommend that you take some of our upper level mycology courses. They're very special and unique to this university. Just another one if you care. All right, and I think that is the end of this video. So I'll see you at the next one shortly.